Hello, hello. And here we are. It is 5 o'clock. It is Tuesday, and therefore it is limiting factors in photosynthesis. Welcome to all of you who are joining in right now in live land, and welcome to you if you're just giving this video a go to try and get your head around limiting factors in photosynthesis. Lovely to have you with me. Just so that you're aware, this is looking pretty complicated because this is what I'm going to be going through this afternoon. Worth you knowing that all boards except for Edexcel A, as far as I can gather from their syllabus, require you to know about the effects of light intensity, carbon dioxide concentration, and temperature on photosynthesis. Eduqas also does not seem to require you to know about any specifics. So Edexcel A and Eduqas don't seem to require you to know about specifics. All other exam boards do. Eduqas only talks about you needing to understand the concept of limiting factors without specifying any. So let's go through what everybody needs to know. AQA, OCR A, Edexcel B, and the Cambridge International Exam Board require you to know about the effects of these three on the rate of photosynthesis. OCR A also require you to know about the effects on the Calvin cycle. And OCR A also has to know about water stress. OCRB actually don't need to know about all three of these. They only need to know about light intensity, temperature, and then these three down the bottom: light wavelength, light duration, and pH. And finally, in addition, AQA and Cambridge International Board need you to be able to apply the knowledge of limiting factors in photosynthesis to how they could be used in agriculture. So I'm just going to spend a few minutes going over that. We're going to go through this in pretty much this order. So hopefully it will be very straightforward, and up in the corner here, I am going to say exactly which exam boards need to know what is on the slide. So let's kick off and do a little bit of context. So limiting factors are factors that are restricting the rate of photosynthesis. I'm going to do a definition in a minute. And some of them specifically affect the light-dependent reaction, and some of them specifically affect the light-independent reaction. I'm going to go through that too. And just to put this whole lot kind of into context and make it a little bit relevant to every day, the light intensity, either in the early morning or at dusk, may be the thing that is limiting photosynthesis during the daytime, particularly at dusk. And I say particularly at dusk because, of course, by the end of the day has kind of warmed up and still got the warmth, but at dusk the light intensity might have increased. Obviously, in early morning on a winter's day, you're going to have temperature in there as well, competing to be the limiting factor. On a cold day, temperature could be the limiting factor that just slows all photosynthesis down. It's why plants don't grow fast in the winter. There's a surprise. It's not because of lack of sun generally, because we can have nice sunny days up here in Europe, but we might not have the temperature. And finally, on a warm, sunny day, it's not going to be either of these two that are slowing things down. It's quite possible that it will be the carbon dioxide concentration in the air. So I'm hoping that just puts this a little bit of relevance around this topic, rather than it just being some theoretical bit tacked on the end of photosynthesis. So here is your definition. The limiting factor is the factor, because it will only be one. It will only be one that determines the rate of photosynthesis. I personally don't like the word limiting because it kind of says that it's stopping it. It does decide it. It does determine it. And limit is a perfectly okay word for you to use. What you shouldn't use in an exam is the word inhibits or reduces. Because of course, if the amount of limiting factor is increasing, then the rate of photosynthesis is going to increase as well. So clearly these words aren't appropriate to use, but sometimes people use them kind of thinking that they mean the same as limits, but they don't. So just be careful about that. And here's the key thing about the limiting factor. It is the one that is in shortest supply at that moment in time. And the best analogy I can give you is a car assembly plant, because a car assembly plant is lots of parts of the process happening one after the other, which is a little bit like photosynthesis, with each bit of the reaction, whether that be light dependent, or light independent, each bit of the reaction working one after the other. Whatever is in shortest supply is going to decide how fast cars are produced. So let's pretend there are 3 processes in a car assembly plant. The windscreen gets put on first, the wheels get put on second, and then the car is painted. If there is a lack of windscreens, it is going to be the lack of windscreens that determines how many cars roll off the production line at the end of each day. If, however, you've got plenty of windscreens and plenty of tyres, but the guy who does the paint job on the car is on a go slow, that is going to be the limiting factor in the process. It's how quick he paints the cars that is going to be how fast the cars come off the production line. In any process where there are a series of steps, by definition, the step that is the slowest will be the one that determines the speed of the whole process. And that is what is happening here in photosynthesis. There is only one factor, and it's the one that is in shortest supply that is going to determine the rate of photosynthesis. So let's go on to think about light intensity. Now, light intensity affects directly the light-dependent reaction. But it is going to affect the light-independent reaction because the light-dependent reaction produces ATP and it produces NADPH, both of which are needed for the light-independent reaction. So if the light intensity decreases, there is going to be a decrease in ATP and a decrease in NADPH. And there might be a decrease in oxygen. But just remember that oxygen is a waste product. The plant couldn't care less about it. It doesn't need it for anything. And it's certainly not needed in the light independent reaction, i.e. the Calvin cycle. So here is your classic graph for the effect of light intensity on the rate of photosynthesis. Remember that here is really dark and here is bright light. And if the light intensity is 0, then so will the rate of photosynthesis be 0. And that's important because at low light intensities, the rate is directly proportional to the light intensity, because when one is 0, so is the other. And notice here that I have highlighted where light intensity is the limiting factor. Because if light intensity increases, so does the rate of photosynthesis. And equally, if light intensity decreases, again, so does the rate of photosynthesis. But above a certain light intensity, by about here, for example, at this light intensity, the rate of photosynthesis is constant. And that means that another factor is limiting the rate of photosynthesis. And this single experiment isn't telling us what that is, but we can just say another factor. And if you were talking about this in the exam, I would go that extra step and suggest another factor. For example, carbon dioxide or temperature. Oh, thank you, Ibrahim. Okay, you want C3, C4 in CAM plants. I'm guessing you're on the on an international board. Not many UK boards need that, but I can look into doing that for you. So this is only required by OCR A. Here is the effect of light intensity on the Kelvin cycle. And here is a quick reminder of the Kelvin cycle and therefore the effect of light intensity. Because of course, if there is little or no light, there is going to be no NADPH and no ATP. And hopefully that immediately tells you which parts of the Kelvin cycle are really going to be affected. So I'm hoping this graph will make sense. When the light is switched off and the dark period happens, the amount of GP, the relative concentration of GP, glycerate phosphate, increases. And that's because it's kind of accumulating because there's no NADPH or ATP to turn it into TP. Oh, yes, yeah, here's my reminder that ATP and NADPH is made in the light-dependent reaction. So here's my explanation about why GP rises. And hopefully it also makes sense that TP decreases because there's no NADPH and ATP to turn any GP into TP. And therefore, and also the TP that was there to start with is getting turned into sugars. So the amount of TP decreases in the dark. And the amount of RuBP decreases because there is no ATP for TP to be regenerated into RuBP. So hopefully this makes sense. And once light is switched back on again, of course, ATP and NADPH are produced, and therefore this Kelvin cycle can carry on as normal. So let's now move on to the effect of carbon dioxide concentration on the rate of photosynthesis. This directly and really only affects the light-independent reaction, the Kelvin cycle. Just worth you knowing, and you probably should know this from GCSE, that the concentration, oh, and can I just say, these square brackets are shorthand for concentration. It's a really common convention in chemistry to, if you've put the square brackets around a substance, it means the concentration of the substance. Really useful to use this in any revision cards, I think. So the concentration of carbon dioxide in the air is about 0.4%, and you probably should have that figure in your mind. So here is again the classic graph for the effect of concentration of carbon dioxide on the rate of photosynthesis. And again, logically, when there is no carbon dioxide, there is zero rate of photosynthesis. And again, that means that in these low concentrations of carbon dioxide, the rate of photosynthesis is directly proportional to the concentration of carbon dioxide. And once again, I have highlighted where the carbon dioxide concentration is a limiting factor. And you can provide evidence from the graph if the exam question asks you to, as you did with light intensity, because as carbon dioxide concentration increases, so does the rate of photosynthesis. And conversely, as carbon dioxide decreases, so does the rate of photosynthesis. And like light intensity, above a certain carbon dioxide concentration, the rate of photosynthesis is constant. It makes no difference how much you increase the concentration of carbon dioxide. And of course, that means, as with light intensity, there is another factor which is limiting. But again, because this is the sole experiment that we've carried out, we can't tell from this graph what is the limiting factor. So the effect on the Calvin cycle. Well, clearly, if there is no carbon dioxide or very much reduced carbon dioxide being fed into the carbon cycle, I'm hoping that you can see that there's going to be a reduced production of GP because carbon dioxide has to react with ribulose bisphosphate to make GP. But equally, there will be an accumulation of RuBP because there's no carbon dioxide for it to react to, so that it is turned into GP. So with decreased carbon dioxide concentration, there's decreased GP because there's no carbon dioxide to make it. There is decreased TP because there is no GP with which to make your TP. And there is an increase in RuBP because there is no carbon dioxide to react with it to make GP. Sorry, my, well, almost typo, if only I typed. So there is no carbon dioxide to react with it to turn it into GP. And therefore RuBP accumulates here. So now let's go on to talk about limiting factors in photosynthesis and the effect of temperature. So once again, this is required by all of these exam boards, and this affects the light-independent reaction directly. Because the Calvin cycle, the light-independent reaction, is an enzyme-controlled process, an enzyme-catalyzed process. And therefore, if enzymes are affected by temperature, then any or metabolic pathway will be affected by temperature. And here is your graph for the effect of temperature. This is the effect of temperature on enzymes, and therefore it's the effect of temperature on the rate of photosynthesis. Notice that the optimum temperature for plants is probably several degrees lower than it is for any animal. It's probably around 20 to 30 degrees, something like that, obviously depending on where the plants are native. At lower temperatures, the rate is proportional to the effect of the rate is proportional to temperature. And actually, in this portion of the graph, this is where temperature is the limiting factor. You can tell if something is the limiting factor because it will be the thing on the x-axis, and the line showing the rate will be going up. So here we have temperature as the limiting factor. And that is because its temperature increases the kinetic energy of molecules and enzymes and causes those enzymes and molecules to collide, and therefore the number of enzyme substrate complexes to increase, and therefore for the rate of reaction to increase. Above the optimum temperature, enzymes of course denature. Can I just point out that if I say that this is the optimum temperature, this is where enzymes denature, but notice that enzymes don't denature below the optimum temperature. It is simply the kinetic energy, the reduction of the kinetic energy, that is slowing down the reaction. I haven't included, thank you, I haven't included light compensation point here. I could talk about it a little bit in the any questions section. the light compensation point is the time at which the rate of respiration is exactly equal to the rate of photosynthesis. In other words, the amount of carbon dioxide used in photosynthesis equals the amount of respiration, the amount of carbon dioxide produced in respiration. But we can have a little look at that in the Any Questions section. So let's talk about the effect of temperature on the Calvin cycle. And so not terribly surprisingly, it does have an effect, as I've already mentioned, but OCR A just need to be aware that this process can occur, photorespiration. Weird, but true. And this is where instead of carbon dioxide reacting with RuBP with the enzyme Rubisco, reacting with Rubisco, being catalyzed by the enzyme Rubisco, instead of carbon dioxide binding with RuBP in the active site, oxygen does. And that means that there is reduced carbon dioxide being fixed into GP. And that means therefore, that means there is going to be less GP and less TP produced. There will be an initial accumulation of RuBP because it is being held back because it can't react with carbon dioxide. But in the end, the amounts will go down because it isn't being regenerated by TP, because no TP is being made. Just as a little aside, and you don't need to know this for A-level, photorespiration is kind of the worst of both worlds, because photorespiration uses oxygen, it gives off carbon dioxide, but unlike photosynthesis, it doesn't produce any sugars, and unlike respiration, it doesn't produce any ATP. So frankly, seems to be a bit useless, and therefore, not surprisingly, reduces the rate of photosynthesis because, of course, it's stopping carbon dioxide being combined with ribulose bisphosphate to continue the Calvin cycle. Now, here is the graph that you might be shown in an A-level exam question, and you need to be able to decipher what is the limiting factor and where it is. So I'm going to go through that. So here we have a graph showing light intensity increasing, but several experiments have been done under increasing light intensity conditions. And these several experiments have been done at low concentration of carbon dioxide and lower temperature, low concentration of carbon dioxide and higher temperature, high concentration of carbon dioxide at the lower temperature, and finally, high concentration of carbon dioxide at the higher temperature. So the question is, where on the graph is light intensity the limiting factor, carbon dioxide concentration, temperature, or something else? So let's go through them. Light intensity is the limiting factor anywhere where the lines are going straight up, but they are not yet plateauing. Because, and the proof of this is that as light intensity increases, so does the rate of reaction at each of these particular conditions. So definitely light intensity is the limiting factor on all of them down here, but you can see that light intensity is the limiting factor in this particular set of conditions right up to this point. So where is carbon dioxide the limiting factor? Well, let's just unwrap these two lines a little bit. we can see that between these two lines, the temperature has increased, but the carbon dioxide hasn't. And to be quite frank, there has been a negligible increase in the rate of photosynthesis. And therefore, the only place, the time or the place on the graph where we can see there has been a significant increase in the rate of photosynthesis is when the concentration of carbon dioxide is increased. So that logically says to me that down here is where carbon dioxide is the limiting factor at both the lower and the higher temperatures when the carbon dioxide is at its low concentration. So you might already be getting the pattern now. So where is temperature the limiting factor? Well, that is here, because we can see we've got the higher concentration of carbon dioxide, but we've got a lower temperature, and therefore temperature is the limiting factor here. And finally, this is where something else is the limiting factor. It's not light intensity, because the rate of photosynthesis has plateaued. It's not temperature, because we're at a higher temperature. And it's not the carbon dioxide concentration. So it must be something else. And that might be, for example, the amount of chlorophyll in the leaves. Hopefully that helps. And finally, let's just talk about this quick application of knowledge of the limiting factors to agriculture. Now, hopefully it makes sense that the only time that we can control the concentration of carbon dioxide, the temperature, and the light intensity is if we enclose our plants. We can't control those things when they're out in the fields. And therefore, it's the use of greenhouses and polytunnels which allow us to control the conditions. We can increase temperature by burning fossil fuels, for example, paraffin. And the added benefit of doing this, and I put that in inverted commas because clearly burning fossil fuels for any reason at the moment is not great. But the benefit is that, of course, if you burn a fossil fuel, it produces carbon dioxide as a result of burning. And therefore, the concentration of carbon dioxide within the polytunnel or the greenhouse will also increase. And that could well be advantageous. Obviously, you can only increase the temperature up to a certain amount. And from an economic point of view, that should be only up to its optimum temperature, because, of course, if you go above the optimum temperature, you're increasing the temperature and risking denaturation. You're going to lose money on both counts because you're going to be putting more energy in, paying for the energy to increase the temperature, but you are going to be decreasing the rate of photosynthesis because of denaturation of the enzymes. John, yes, I taught Jade, indeed. So you can, in a polytunnel and in greenhouses, you can increase the light intensity, and that's by switching lights on or having lights on for a longer period during the day. But again, notice that you only, from the graphs that we've talked about previously, you only want to increase the light intensity up to a certain amount. Because if you increase it anything above that, it's going to make no bogging difference to the rate of photosynthesis. So there is a point above which it makes no economic sense to pay for more lighting, because you're getting the maximum rate of photosynthesis for the least amount of energy that you have to pay for. And finally, the increase in carbon dioxide, you can either do it by burning fossil fuels, by using bottled gas, or, as in the case that I found on the website when researching this, in this greenhouse in Switzerland, they actually remove carbon dioxide from the air and pump it into the greenhouse to increase the carbon dioxide concentration to make the plants grow faster. But notice, just like with light intensity, you only want to increase the concentration of carbon dioxide up to a certain point, because there is no point in paying for the concentration to be higher when that rate of photosynthesis won't change. I hope that makes sense. Unjaded Jade was, yes, not surprisingly, one of the brightest students, because she knew how to learn, she knew how to, or she does know how to learn, she knows how to revise, and she knows how to analyze exam papers so that she can maximize the marks that she got in exams. So yes, she always did do well. But you probably have also seen the Instagram clip, I think, that she put out there, or YouTube clip that she put out there after one of her first A-level exams, where she was absolutely distraught because the exam board had decided that year to add in a load more lines in their answer spacing. And her, along with a number of other very bright students that I was teaching, were very upset because, of course, they were bright enough to fill the lines, and therefore that completely screwed up their timing, which was more than a little bit disappointing and worrying. Do I recommend flashcards? Do you know, John, what I'm going to say to you is have a look at my, I'm sure it's on the YouTube channel, my live stream of how to revise. That will help you. I don't particularly recommend flashcards or notes or anything else, but there is, I do talk about using, I can't think what it's called, graded repetitions. So in other words, you repeat what you need to learn, revising it. You repeat it, intervaled repetition, where you repeat it very shortly after you first learnt it, then you make that interval longer and then longer again to get it into your long-term memory. And one of the ways of doing that does use flashcards. So have a look at my how to revise video or live stream on YouTube. Just want to do a really quick advert for my website, where I have my full complement of videos, which I'm selling for what I'm genuinely hoping is a nominal amount, because my absolute aim is to help students who do not have the money for private tuition. So to subscribe to all of my videos for a year is £50. If you only need the AS videos, it's £25. And here's the fantastic thing, that if you do not have the bank of mum and dad to hand, you can earn 10% commission on friends' purchases and therefore potentially make a flipping profit on your subscription. And there is also a freebie there, how to improve your grade up to an A star, where I also talk about revision, John. So I hope that's helpful. And these are all of the videos that I have up for year one. This is obviously AS topics, and here are the year two topics. And you can see that a whole load of them are coming soon. I just need to get them edited, and they'll be up on the website. And and these are all of the ones that I've added since September. So I think I can reasonably be sure that I'm going to finish the whole lot by Christmas. And whatever I put, let's get back to this. If there is not enough water, this is just logic. Well, I say just, that was a bit silly, patronizing. This is logical. So if there is not enough water in the plant, cells, now here's the thing, especially guard cells, lose their turgidity. They plasmolyze, they undergo plasmolysis. And therefore, because of the particular shape of guard cells, because they're kind of like two bananas, aren't they, joined together? Yeah, oh, a bit dodgy. Here, they're thickened on this edge. That is their adaptation. And therefore, when they lose turgidity, they close. And as a result of that, that means that there is a decreased CO2 uptake because less gas exchange can occur. And as a result of that, photosynthesis decreases significantly. I hope that is relatively straightforward in terms of the logic. But here's the tricky thing. Because plants are constantly doing a balancing act with whether their stomata are open or closed. They obviously want their stomata, and their stomata do open under conditions of high light intensity. There is a mechanism by which guard cells open under high light intensity. But of course, that means that they're going to lose more water because the stomata are open. And so there is a constant balancing act occurring. But the bottom line is that if too much water is lost from the plant, the stomata will close, and therefore photosynthesis will be very much reduced. Oh, thank you, Campbell. Oh, I love biology. It is just a brilliant subject. Yeah, no, Julian, thank you. Lovely to see you, Julian. Well, I wish, oh, if only I could see you. No, that's the weird thing. Because of guard cells adaptation and them being thicker, now I can't do it with my hands, thicker on this inside edge, it actually means that when they lose water from them, they flop closed. Whereas it's probably easier to think about that when they are turgid, when they are full of water, this edge doesn't, oh, no, I can't explain it well. No, I've lost the wording for explaining this. I think you probably do, and I'm not going to get hung up about it. Because they are thickened on this edge, it just means that when they are turgid, they open the stomata. There is a hole here between them. And when they are flaccid, or when they have plasmolized, they go shut. They close. Sorry, bad English. Okay, finally, OCRB. No, the videos on my website, John, you need to pay for, and they are only for your personal use. I make that clear on the website. Look under the frequently asked questions. If you do read the website, I have deliberately worded it so that it kind of is really easy to understand. I am not trying to hold anything back. It is, what you see is what you get. So do have a read of the website, and I'm really hoping that it clarifies things for you. Oh yes, Bethan, balloon and tape, brilliant. Yeah, that's right. It's a fantastic demonstration. Thank you for that. If you are particularly one of the long balloons, I'm assuming, if you put tape down one side and you blow it up, it forms the shape of a turgid guard cell. Yeah. And when there is not enough air in it, it just goes straight, and therefore there is no hole between the guard cells. Yeah, thank you, Bethan. Good prompt. Limiting. So finally, OCRB. Can I just say, all of these differences between the specification, you could easily have a question on anything that I've talked about here, because the exam board would deem it as application of the knowledge of limiting factors. So, it's not a bad thing for you to watch all the way through, just to kind of say. So light wavelength, hopefully you'll remember that white light, the light that comes from the sun and that comes from the lights inside our house, have the full spectrum of wavelengths of light. White light contains all the colors of the rainbow because the colors are light of particular wavelengths. So, and hopefully you can see from here is that photosynthetic pigments, chlorophyll and all of its mates, particularly absorb light at the blue end and at the red end. And therefore it makes sense that we can use a specific color or wavelength of light. And that means that you can put a filter over a white light, because the filter, if you put a red filter over a white light, only red light is transmitted through the filter. In other words, only red light passes through the filter, and therefore only red light will hit the plants. So it means that you can illuminate plants with a particular wavelength. But here's the but. Obviously, if you put a dark red filter over light, it's also going to decrease the light intensity. And that could also very easily occur with blue or purple filter. Hopefully that kind of makes sense, and it's worth noting that. Just to say, and if you've seen enough of my videos, you'll have seen me explain this. Notice here that there is a dip in absorption of the light wavelengths where it is green and yellow. And that's because those wavelengths of light are reflected off the leaves and into our eyes. And that's why we see leaves as green, because these wavelengths of light are the one set of wavelengths that are hardly used in photosynthesis. It's the wavelengths at either end of the spectrum that are used primarily. I mean, obviously some is used, but not as much. Light duration, only OCR B need to know this, is that dear old plants need a rest. The optimal duration of light that you can have plants illuminated for, picture those greenhouses with the lights on, the optimal duration is about, I think I've said, is it 16 hours. Much more than that, and actually the rate of even if you've got the perfect light intensity, the rate of photosynthesis will go down. It's almost as if the plants are getting tired, dear of them. And therefore, it's just worth kind of recognising, OCR B students only, that a dark period is required for when you put on the light at that perfect light intensity for the photosynthesis to be at its maximum. I hope that makes sense. And we have now finished the live stream on limiting factors in photosynthesis. I was not expecting that to be as long, but when I started looking at all the specifications and realizing how they were just subtly different one to the other, and me knowing that you would all want to know exactly what is included, that's what has taken the time. But all except for the I've just realized I haven't done pH. Really sorry. Obviously something's gone wrong here. Sorry about this. Let's just do this quickly. pH. This is also, it affects the light independent reaction, because the light independent reaction, the Calvin cycle, is an enzyme catalyzed reaction. So just as pH affects enzymes, it affects the Calvin cycle. And if pH is too far from the optimum, and in this graph, here is the optimum where the rate is at its maximum, enzyme denaturation occurs. And therefore the rate of photosynthesis decreases. I have now finished. And I'm sorry, that was a